Thank you, everyone. I want to thank the organizer for giving me this opportunity to tell you a little bit about regenerative patch technologies and the technology that we are developing as a bioengineered implant for the treatment of age-related macular degeneration. First, let me tell you a little bit about the company, about Regenerative Patch Technologies, or RPT as we call it. It's a clinical stage company, and it was formed as a consortium of 4 different universities: USC, University of California at Santa Barbara, Caltech, and the City of Hope. And it came together to form this implant technology that is being used for our lead product, which is for the treatment of advanced dry AMD. It's a huge opportunity because that's a tremendous unmet medical need. And RPT has raised approximately $42 million in non-dilutive funds from the California Institute of Regenerative Medicine to fund development of this particular product through the IND phase and through our phase 1/2A clinical trial. RPT has also secured equity financing through Santen Pharmaceuticals through its venture arm, Santen Ventures. Also, our phase 1/2A clinical trial is complete, and there's 1-year follow-up on all patients. And we're preparing to initiate our phase 2B clinical trial next year. We have FDA clearance and agreement on that trial design for the phase 2B and the endpoints to be used in that study. And we also have issued and pending worldwide patents that essentially cover every aspect of the implant and the tools that are used to implant this particular device. So what's the clinical problem that we're trying to address? It's the dry form of age-related macular degeneration. And age-related macular degeneration affects 1 in 8 people over the age of 60. There are over 10 million people with the dry form of AMD in the US, and 1.7 million people with the advanced dry form in the US. And what does it lead to? It leads to blindness, inability to read, drive, recognize faces, a tremendous loss of independence, increased falls and injuries, and dependence on caregivers. In fact, all the patients in our clinical trial were dependent upon caregivers to get to their particular visits and follow-up visits. The tragic thing about AMD is that there are no approved therapies for this disease. Conventional drugs have failed, and most drugs that are currently in advanced development are drugs that are proposed to simply delay the progression of disease. And we're looking at developing an implant that has the potential for, in fact, improving vision in these patients. So let me tell you a little bit about the pathology of dry AMD and how our product actually fits into this. This implant is really designed to address the disease pathology in geographic atrophy, which is the most advanced stage of age-related macular degeneration. As macular degeneration progresses, you get a loss of the retinal pigmented epithelial layer and a Bruch's membrane, the Bruch's membrane upon which this epithelial layer sits, and causes it to polarize. The loss of this structure, the RPE cells on the Bruch's membrane, causes then the subsequent degeneration of the retina photoreceptors above it, and subsequently the retina. So it is really the loss of this particular structure, this tissue structure, that can be catastrophic in patients with dry AMD. And our approach, really is shown on the right-hand side, is to make an implant in which you have polarized RPE cells on a synthetic Bruch's membrane that is inserted in the subretinal space that can serve as a support for the overlying photoreceptors and the rest of the retina. And it acts to provide a series of neurotrophic factors, and also provides metabolic support for the entire retina. So this slide shows just pictures of the implant. They are polarized retinal pigmented epithelial cells that are on this ultra-thin perylene membrane. Pictures here on the left-hand side, the pigmented epithelial cells that we derive from human embryonic stem cells. In the middle is a picture of the ultra-thin perylene membrane. The cells actually grow on the bottom portion of this membrane. It's also lined with a mesh that you can see forming circles on this ultra-thin membrane, which allows for handling of the whole implant. On the right-hand side, again, you can see pictures of the entire implant. It is about 3.5 by 6.5 millimeters in surface area, and therefore essentially covers the whole macula. It is a one-size-fits-all type of implant. It also has a handle that is used to insert the implant into the subretinal space, and it has a little diagram on there, or a little feature called, what we call it, the Snoopy nose, which provides directionality, in which the surgeon can actually tell in which orientation this person is implanting in the subretinal space. So the implant has two very important components. There's the RPE cells, which are the biological component of the implant. They're derived from pluripotent stem cells. They polarize as native RPE cells do. And they produce what is shown on this left-hand side picture, microvilli that interdigitate with photoreceptors to provide nutrients, to provide the removal of waste products, and also to provide trophic factors. These RPE cells also produce something called RPE65, which is a very important visual component, cycle protein, a very important vision protein, important for creating the perception of vision. The second component is an ultra-thin perylene membrane. And the ultra-thin perylene membrane, about 0.4 microns thick, and it's composed of perylene, a monomer. It's this USP class 6 monomer. So it's highly biocompatible and can be used, has been used in implantables for the last 30 years. The important feature that was discovered by Y.C. from Caltech is that if you can manufacture this perylene membrane to a specific thickness, about 0.4 microns thick, what you can find is that this membrane, the diffusion constants and diffusion characteristics of this membrane will mimic that of the Brooks membrane. And hence we call it a synthetic Brooks membrane. The other thing that's very important is that this perylene membrane, in fact, is foldable. It has memory so that when it unfolds into a flat shape. So this folding property of the membrane and of the implant allow it to be curled up into a very small diameter such that it can be implanted very easily. So we're really thinking about this implant as being a tissue replacement therapy, replacing the RPE layer, replacing the degenerating Brooks membrane that are found in patients with dry AMD, and particularly in geographic atrophy. And that has really implications for our development path for the product. So if you look at traditional drugs, they're looking at slowing the progression of disease. Hence, many patients are required in clinical trials in very long developmental time frames. However, we're looking at improvement of vision. So that essentially allows us to look at decreased numbers of patients and shorter development times, because we're looking at shorter follow-up times also for patients to look for improvement in disease. So how is this implant produced? It's produced in two different phases. First, there's the production of the RPE cells. They come from GMP manufactured master and working cell banks of embryonic stem cells that are subsequently differentiated and expanded to form intermediate cell banks. And these intermediate cell banks are cryopreserved. They're RPE cell cryopreserved banks. For the second step of the process, what we have is that the vials from the intermediate cell bank are plated onto the cells in those vials, are plated onto the ultra-thin membranes, and then they're subsequently cultured and subsequently processed. So essentially what we have are GMP manufactured working, master working and intermediate cell banks. There are no animal-derived products used in this manufacturing. The GMP manufacturing has been, in fact, successfully used now at the City of Hope for the manufacturing for our phase 1/2A clinical trial, and it's being transferred now to a commercial CMO for future development. The process is amenable to automation, which is very attractive from the perspective of the number of patients that you would have to consider treating during commercialization. And an important feature now is that we can cryopreserve the final implant. The final implant is cryopreserved, will be thawed in the OR, and subsequently implanted directly. So again, the manufacturing in this is ready for phase 2B clinical trials. Just some data here to show that even the cryopreserved and thawed form of the implant remains polarized. You can see here by looking at various apical and basal markers and looking at confocal microscopy that these cells, in fact, have a different apical and basal surfaces, very similar to what you would expect to see for non-cryopreserved RPE cells. And they do form these villi, as can be seen on the right. They still form the villi that can interdigitate with photoreceptors. So how do we implant this device? We've developed a custom tool that can be used to take up and curl the implant, and then subsequently inject it into the subretinal space and unfurl that implant. So these pictures depict what happens. There's a vitrectomy. There's then a subsequent hydrodissection of the area of geographic atrophy from the retina, detachment of the retina. There's a little retinotomy about 1.5 millimeters that is made in the retina to allow for the transposition of our membrane through the retina. So the implant is taken up by a tool, curled in the shaft of that tool. That tool is put in through the retinotomy into the subretinal space, and it is then ejected. It is and unfurls. There's subsequently a tamponade in order to reattach the retina. This is administered as an outpatient, takes about 2.5 hours, and it uses well-established procedures that retinal surgeons are very, very trained in. We've now completed a clinical trial, completed enrollment in a clinical trial, a phase 1/2A clinical trial, which was a single arm open label study in patients with geographic atrophy. There were 16 subjects that were enrolled. All patients in this trial were legally blind. So they had less than 2200 or worse vision. And we treated the worst eye, the worst vision eye. Again, it's one size fits all, so we did one implant. Patients were given a 68-day course of tacrolimus, 8 days before implantation, and tapering at day 42 and completing tacrolimus at day 60. And the endpoints, primary endpoint was obviously safety and tolerability 1 year after implant. This was conducted at 6 different clinical trial sites in California and in Arizona. All of the implantations were done at the University of Southern California. So there are 3 things I really want to talk about in terms of the clinical data. First is delivery. We have delivered this particular implant. There were no safety concerns associated with the delivery of the implant. You can see, in fact, that the implant, in this particular picture, the upper left-hand corner, you can see that the area of GA in this particular patient, and that it's days 28 through 65, you can see the position of the implant over the area of geographic atrophy. Some important features were that We were always able to target the implantation over the area of geographic atrophy. The implant was stable over time. There was no evidence of any degeneration of the implant, no movement of the implant. There was no apparent rejection, obvious rejection of the allogeneic cells that are on this particular implant. And again, this was using techniques that were very familiar to retinal surgeons. With regards to the safety of the implant, we saw no unanticipated serious adverse events due to the surgery or the implant. There were three patients, in 3 of the first 7 patients in the study, there were ocular SAEs that were related to hemorrhage and edema that were reported during the first year of the study. What we determined was that some of this hemorrhage and edema was due to the lack of hemorrhage control during the implantation. That was rectified, and subsequent 8 patients had no reporting of SAEs due to hemorrhage or edema due to this better hemorrhage prevention. The 68-day tacrolimus immunosuppression was tolerated well in most patients. There was only 1 patient who had to be taken off of the immunosuppression at day 42, towards the end of the immunosuppression period. Otherwise, everyone tolerated it well. There was no evidence of rejection of the allogeneic cells, even with very short immunosuppression protocol. And one of the things that was important to show was that in this case, we have a one-size-fits-all implant. And in some cases, the implant might be going over areas where there's relatively normal retinal architecture. So even in the cases where there was normal retinal architecture, as you could see on the left-hand side here, that there was good preservation of that normal retinal architecture, even when the implant was placed over an area with intact host RPE cells. Again, a very important safety feature. So what about activity? Did we see any activity of the implant? In this case, what we've looked at, looking at best corrected visual acuity, we've also looking structurally at the implant and the retina over the implant. In some of the cases, we've seen the reappearance of the external limiting membrane over the area of geographic atrophy where it was missing beforehand, suggesting that there might be some sort of restructuring, remodeling of the area of GA over the implant. We've also looked at best corrected visual acuity. And in this case, all of these patients were legally blind. And we saw that 67%, or 10 out of the 15, either remained stable or improved in vision. 27%, or 4 out of those 15, actually showed a greater than 5-letter improvement in visual acuity. And in fact, the improvements range anywhere from 6 letters to 13 letters. So you can see on the left-hand side what this translates into. Here's the standard eye chart, EDTRS eye chart. And in this case, I'm showing you the eye chart for someone who has severely impaired vision. So this eye chart is placed 1 meter away from the patient, unlike for normal sighted people where it's placed 4 meters away. So for 1 meter away, patients in our clinical trial, you can see the baseline arrow. This is what they could serve. They had 2400 vision and could visualize this ONVS line at 1 meter away from them. However, after 1 year, you can see how their vision improved. So this is very important, especially for patients with very severe impaired visual acuity. And these, again, the signals of efficacy are very encouraging for the product. So where are we now? We are in the process of designing and preparing for a 3-stage phase 2B clinical trial that has a lead-in open label stage for new surgeons on the trial, a randomized assessor mass stage, which will be the heart of the clinical trial, comparing the controls to patients who have received the implant, and then a subsequent control subject open label phase, which those patients who are in the control group are in fact allowed to receive the implant. All patients will be followed for 12 months for primary outcomes and an additional year for safety. The endpoints have been cleared as registration endpoints by the FDA, and we do have our pathway for a BLA program. So I'd like to stop here and summarize. Regenerative patch technologies developed an implant as a tissue replacement therapy for advanced dry AMD. The implant has been produced and has been able to, manufacturing has been able to support phase 1 and now phase 2 clinical trials. We have a cryopreserved form of the implant that will supply global distribution. We're about to, we have our phase 1/2A clinical trial, which has had a good safety profile and good efficacy signals. And we're in the preparations for our phase 2B clinical trial. And I think I'll end here by thanking patients, their caregivers, and all of the various teams that have been involved in this program. Thank you very much.